everybody! So today we're going to talk about genetics, which is chapter 14 in our AP Biology book. So as I go through the PowerPoint, I would like for you to be taking notes on the note paper. This is what it's going to look like. It is this Word document that I put also in Schoology along with a copy of this PowerPoint. Okay, so let's get started. Genetics is usually a favorite topic for a lot of students. It's a lot of fun trying to figure out like the probability of the outcome of offspring. There's a little bit of math involved. However, it's not so intimidating because it's actually interesting. It's about genetics. We're going to be talking about genetics of pea plants first, and then we're going to go into human genetics later on. Okay, so the guy that is known as the father of genetics, let me just move my face so you can see his face, is Gregor Mendel. So Gregor Mendel, he was a monk. So he was a priest, and he had a lot of time to pray, and he loved math, he loved botany. So he combined those two passions. With all the time he had on his hands, he combined those two passions of math and botany together, and he studied pea plants. And as a result of his 12-year-long study of these pea plants, he discovered a lot of patterns of heredity that we're going to be learning about in this chapter. He is considered the father of genetics mainly because he was the first person to explain some of the patterns of inheritance that we'll be discussing, like dominance and recessive, genotype, phenotype, and he even came up with these two laws of genetics, the law of segregation, the law of independent assortment, which we'll be talking about in this video. So what was he looking at? He was looking at sweet pea plants, and it was a 12-year-long experiment with over 10,000 pea plants. Like I said, he did have a lot of time on his hands, and it was his passion, math and botany. So this is something he enjoyed doing. And so sweet pea plants are not edible. They're more, I guess you could say, ornamental plants. They grow in the garden, and they have a wonderful floral, sweet smell. I mean, when you go to like Victoria's Secret or Bath and Body Works, they're going to have lotions and candles with the sweet pea scent. So that's something that some of you may be familiar with. So he studied 7 heritable pea plant traits. He was looking at seed shape, like you have round versus wrinkled seed shape. You have seed color yellow versus green. You have flower color like purple versus white. Pod shape, inflated versus constricted. Pod color like green and yellow. And then you also have stem length, like long stem versus short stem. Flower position, like axial versus terminal. What does that mean? It's just, where is the flower found on the stem? Is it found along the stem? That would be axial. Or is it found at the end of the stem? That would be terminal. I'm already lagging. I don't know what's going on with my computer, but it's okay. I'm just going to continue on. Okay. It is a little bit weird to like hear my voice and my mouth don't match up, but what can I do? So these are like the 7 characteristics that he studied in pea plants. And again, this is why it took so long as well, because he was studying these characteristics. There's 7 of them, and it took him about 12 years to study these. We did talk about this in class earlier, about how the flower is the sexual organ of a plant. And you have male parts and you have female parts, and both male and female parts are found on a flower for this sweet pea plant. And now I'm reminded of something that somebody said in period 1, which I'm not going to repeat here, but it has something to do with flowers and smelling, the scent of flowers, but we're not going to go there here in the video. But it's truly, it makes you think of flowers in a different way. So anyways, with Mendel, he used 2 basic tools. He used scissors and a paintbrush. So as I was saying, flowers are the sexual organs of a plant. they have both male and female portions. And maybe I should just quickly draw it here because for those of you that didn't hear me in class, so you have at the center part of the flower, you have what we call the carpel. That is the female portion of the plant. Okay, so I'm going to draw the female symbol. And in the middle of the carpel, at the very base of the carpel, you have these eggs. Okay, just like how eggs are stored in ovaries. This is called the ovule. Okay, the ovule, which is part of the carpel, which is the female portion of the flower. And then you have the petals. The petals obviously are to attract pollinators. That's what just amazes me about plants, is like every part of the plant has, it's there, it has a purpose. Okay, it has a purpose. And in this case, it has petals to attract pollinators because out of the male parts of the flower. So here we have, this is called the stamen, and the stamen grow all around. There's usually lots of stamen that grow all around that main carpel. And at the tip of the stamen, and I'm going to draw the male symbol for stamen, and at the tip of the stamen you have the anther. And the anther produces pollen, and the pollen grain contains sperm. And so we were saying in class how if you are allergic to pollen, you're allergic to plant sperm. That's what you're allergic to, okay? So the pollen is basically little packets of sperm. They will land on the top of the carpel, and then once they land on top of the carpel, a pollen tube will form, which is going to dig down to the eggs, fertilize those eggs. And once those eggs become fertilized, you get a little embryo in a seed. Or in this case, with the sweet pea plants, the embryos inside of the peas. So going back to Mendel's experiment, what he wanted to prevent was self-pollination. Many plants, many flowering plants are able to self-fertilize, so they can fertilize their own eggs. Here, and now you can see that in my drawing that I drew, the pollen grains, the plant sperm landing on its own carpel and fertilizing its own eggs. So what he wanted to prevent was self-fertilization or self-pollination initially. So he cut off the stamen, basically castrating the flower. Okay, the purple flower, he cut off the stamen, and then he took from another plant, another sweet pea plant that bore white flowers. He took a paintbrush, he gathered some of the pollen or the plant sperm onto the paintbrush. He cross-pollinated. Okay, he applied it to the carpel of the other purple plant. So this is what we call cross-pollination, where cross-pollination, okay, where you are transferring sperm or pollen from one plant to another plant. Okay, and so now you have the eggs are fertilized, and here you have a pod, a sweet pea pod, with 1, 2, 3, 4, 5 little embryos. Okay, and then what did Mendel do next? He planted the little seeds or the little peas in the ground, the 5, and they grew out of the ground, and he called them the F1 generation. F1, first generation offspring, F1. And I did not mention that the two parents, the purple and the white plant, he called them the parental generation. They are the P generation, the parental, and they produce the F1. And you can see in this diagram or this picture that the parents, one was purple, one was white, and what color are the F1 generation? What color are their offspring? They are all purple flowers. Okay, and so he saw this pattern quite often when dealing with the different characteristics of the pea plants. So flower color is just one of the characteristics that he studied. He also studied pea color, pea shape, pod color, pod shape, like that table I showed you earlier. And he saw this pattern. So for example, the pea color yellow versus pea color green, when he cross-pollinated them, the F1 generation were always yellow. Okay, so he saw this pattern, and again, that's why he is the father of genetics, because from the pattern he was able to come to conclusions about inheritance patterns. And that's what this chapter is all about. So looking at Mendel's work, the P generation, they were true breeding, or you can say purebred, okay? Purebred, purebred purple, purebred white, or true breeding, okay? And he cross-pollinated them. So now you guys know what cross-pollination means, right? Taking the pollen from one plant and applying it to the carpel of the other. And then he produced the F1 generation, and they were all purple. And he called them the hybrids. Why are they hybrids? Because each one, the offspring, has purple gene. And then they have a white gene. So they all have like a purple gene and a white gene, purple gene and a white gene. They're a mix of, they have a purple gene and a white gene. They're not mixing colors, but they're all purple, but they have the white gene in their genome, okay? All right, next what he did was he allowed for the F1 generation to self-pollinate, and self-pollinate, sweet pea plants are able to self-pollinate. And for the F2 generation, the next generation, it was always this ratio or percentage or fraction. It was always 75% purple or 3/4 purple, and then 25% white or 1/4 white. So this 3-to-1 ratio, 3 purple, to 1 white. And I probably shouldn't use that W because later on we're going to be talking about letters that represent dominant alleles versus recessive. But just for now, we'll just say 3-to-1, 3 purple, 1 white. Okay? He saw this pattern over and over again, not just with sweet pea flower color, but with other traits that he studied. Sorry, I'm getting distracted. There's like this squirrel like running around in the backyard. So what did Mendel's finding mean? He was studying how these traits with the sweet pea plants, they come in alternative versions. You have purple versus white for the flower color. You have the pea color, green versus yellow. So these alternative versions, they're called alleles. Okay, alleles. So basically alleles are alternative genes. You have the purple gene, you have the white gene, okay? And here, just like us, sweet peas are also, they're diploid. They're diploid, 2N. Remember, that's a symbol for diploid? 2N or diploid means 2 sets, okay? What do we mean by 2 sets? It means 2 sets of chromosomes. One from, let's say, mom, okay? One from dad, right? One from the egg. Maybe I should just write egg. One from the sperm. Okay, so just like us, the sweet peas are also going to be reproducing sexually. They're going to have gametes. You have the egg, you have the sperm, the egg and the ovule, the sperm and the pollen grain. Okay, and so they're diploid. They're going to have they're going to have 2 genes for every trait. So they're going to have 2 alleles for flower color. In this case, the F1 generation, for the F1, they're hybrids. They're hybrids. They're going to have a purple gene. Okay, so they're going to have a purple gene or purple allele. They're going to have a white allele or gene. So they're hybrids because they have 2 different ones, purple and white. Okay, so they're diploid just like us. They reproduce sexually. For each characteristic, an organism inherits 2 alleles, one from each parent, right? What is an advantage for being diploid, having 2 alleles for every trait? So instead of having just 1 gene for a trait, you're going to have 2. Let's say for whatever reason, maybe this individual has inherited some kind of mutation, okay, a mutation that is going to lead to a disorder, okay? And then they've also inherited a normal version of a gene. So if we have 2 genes for every trait, it just so happens that most mutations, most disorders are recessive, meaning that the normal version is going to like take over. It's going to mask the mutated version. So that person or that individual is going to have an advantage by having the normal gene express itself over the mutated gene. So the mutated gene is not going to show up in the individual. It's the normal one that's going to show up. So you can think of it this way. If you have 2 genes for every trait, let's say you get one that's a bad copy, like maybe a mutated version, at least you have a normal one as a backup. Does that make sense? So if you only have 1 gene for every trait, you're kind of at a disadvantage, because whatever you end up with, whatever you inherit is what you're stuck with, right? But if you have 2 of every gene, being diploid is an advantage, because the chances of you getting a normal one, and you have a better chance of getting a normal gene, and you can fall back on that if you inherited a bad copy or a mutated copy. Okay. So I've been saying dominant versus recessive. Basically, a dominant allele, it's the functional protein. It's the one that masks the other allele. Whereas recessive is going to be masked. It's not going to show up when it shows up in an individual with the 2 different alleles. So let's say that you have purple, and then you have the white. I'm going to write it out because I don't want to confuse us with the letters just yet. The purple gene versus the white gene. The dominant allele is the purple gene. It's going to produce a protein. It's a gene that codes for a protein that's going to give it a purple color. Okay, that's going to give it a purple color. And it's going to mask the white gene. The recessive allele is going to be white. And we say that the white gene is a mutation. We say that it's a mutation, and it's going to make a malfunctioning protein. And that's why it looks white, because white is not a color, right? There's no color with white. So I don't know why I wrote white color, but you know what I mean. It's white. The color of the flower is white. And so it doesn't make the purple protein, the purple color protein. So it's going to be without color. It's going to be a white. It's going to look white to us. So I hope that makes sense. So that's what's going on with the purple versus white. The dominant allele is going to make the functioning protein, whereas the recessive allele is making a malfunctioning protein. It's not going to be purple, so it's not going to have a color. It'll just be white. And also, I did want to say that when we talk about fruit flies in the next chapter, fruit flies, you're going to be hearing this word like wild type. Wild type is the dominant trait. Wild type is like the one that you would see most commonly in the population. It's the one that you would consider to be the normal version of that trait, versus the recessive. The recessive would be the mutated. So you have wild type versus the mutation. Okay, so we're going to be talking about wild type again in the next chapter, but I just want you to know that wild type usually refers to the normal, dominant versus the mutated version. Okay, wild type versus mutated version. So in the case of the pea plants, the wild type is the purple color, and then the mutated color is the white flower. Okay. Oops. So let me erase that so you guys can actually see that. So as I was saying earlier about letters representing alleles with letters, we are going to be doing Punnett squares. And the Punnett squares is going to tell us, like, what is the probability of the outcome for offspring. If you know what the parents are in terms of the genetics, you can do Punnett squares to figure out what's the probability of the parents having an offspring that's going to be purple or white. So we're going to use letters to represent the alleles. The dominant allele, we're going to take the capital letter, the first letter of the dominant allele. We're going to take the capital letter of it, and that's going to be the symbol for the dominant allele. Sorry. So for example, purple is dominant, to white. So the first letter of that dominant trait is purple, is P. So capital P will be the symbol for the dominant allele. All right. The recessive allele is white, right? But we're going to take the same letter, but we're going to lowercase it. Okay. So the symbol for white is going to be little p. We want to use the same letter. We want to use the same letter. The dominant trait will be capital, but then the recessive trait will be lowercase. We want to use the same letter because when we talk about codominance and incomplete dominance, that's when the different letters come into play. But for the genetics cases or genetics problems where we're dealing with dominant versus recessive, we always use the same letter. One's capital, one's lowercase, just so we know what kind of inheritance we're talking about. In this case, if you see uppercase and lowercase, you know that one is dominant to the other. Okay? But if you use two different letters, we're going to be talking about incomplete dominance. Like, for example, red, the symbol for red will be R. The symbol for white will be W. And R and W together make a different color, pink. Anyways, I think I'm going a little too ahead of ourselves. So genotypes. Genotypes are the genes, okay? They're the two genes that an individual inherits from their parents. The two genes. So the two genes, if they happen to be the same, we say that that individual is homozygous for that trait. Homozygous for that trait means that they have the same alleles. The two genes are the same. So this one could be from mom. This one could be from dad. It's the same, two of the same. Purple, purple. Same here. White, white. So it's two of the same genes. The parents gave two of the same genes. So that person, that individual, that flower, whatever we're talking about, is homozygous purple. So this is homozygous purple or homozygous white. Okay? Then you have heterozygous. Heterozygous means two different alleles. So purple and white. So two different. Okay, so that's the genotype. The genotypes are basically the two letters that represent the two alleles that that individual has inherited. Okay, so genotype are the two genes, the two letters that represent what that individual inherited from the parents. Okay, so that's genotype. And then you have phenotype. What does phenotype mean? Phenotype is the physical appearance. Okay, the physical appearance. And then, one way to remember that, and a lot of people remember it this way, is that phenotype starts with PH, so does physical appearance. Okay, so genotype is the genes, whereas phenotype is the physical appearance. Okay, so big P, that's the genotype, but the phenotype is purple. That's the physical appearance. Little p, that's the genotype, but then the phenotype is white. Okay? Heterozygous, big P, little p, that's the genotype, but then the phenotype is purple. So even though, this heterozygous individual is a hybrid, even though it carries a white gene, it's not white in its phenotype. It's going to be purple in its phenotype because Mendel learned that the purple gene is dominant to the white gene. It masks, it covers up the white gene. You don't see it. Physically, it's not expressed. Okay? Oh yeah, and then I had a little picture. I don't know if I wouldn't consider this a meme, but let me show this to you guys. It'll pop up as soon as I erase this here. Right? It's the goats, right? Heterozygotes. Hold on. So heterozygotes, you can see here. So somebody that's a heterozygous, we call them a heterozygote. Okay? So in this case, it's heterozygote, spelled G-O-A-T-S. Whereas a person that is heterozygous, we say, maybe I'll write it up here. They are a heterozygote. They're a heterozygote because they have two different alleles for that trait. But in this case, it's spelled goats, G-O-A-T-S. Anyways, I thought it was cute because it's like half black, half white, two different halves, just allele uneven, right? I thought it was cute. You guys are probably not even like, you're like, whatever, Miss Shem. Okay, probability. Probability is like the likelihood that a specific event will occur. For example, if you flip a coin, you have heads on one side, tails on the other. What's the probability of getting heads, flipping the coin and getting heads? 50% chance, 50-50 chance, right? So this is a Punnett square. Okay, this is a Punnett square. A Punnett square is a large square that's divided into four smaller squares, sorry. And those four smaller squares can give you four different possibilities depending on, you know, the coins that you're flipping. So, for example, let's try to figure out, like, what is the probability of flipping a coin twice? So you're going to flip a coin twice, but what's the probability that you're going to get heads, you know, one after the other, okay? Solution for two coins. So when you flip a coin, it's either heads or tails, okay? So this is like the first flip. The second flip, so let's say you want to flip the coin a second time, what is the chance of getting heads or tails, okay? So you're going to flip the coin twice. Each time that you flip the coin, it's 50-50, heads or tails. Heads or tails. So what is the probability that you're going to get heads and heads? What is the probability that you're going to get heads and tails? Okay? So this heads is that one. And then what is the probability of heads and tails? And then what's the probability of tails and tails? So in a Punnett square, what's going on in the little square is the possibility of getting heads, tails, tails, okay? And the thing about Punnett square is that you can see that it's divided into four squares. So 1 out of 4, I only see 1 out of the 4 is heads. So there's a 1 out of 4 chance or 25% chance that if you flip a coin twice, you're going to get heads. Okay? And then there's also a quarter chance or a 25% chance that when you flip a coin twice, you're going to get tails. Okay? And then for heads, tails, or tails, heads, it really doesn't matter. Heads, tails, or tails, heads. There's a quarter chance here, quarter chance here. You add them together, okay? That's going to give you half chance, a 50% chance. A 50% chance that you're going to get heads, tails, okay? Or tails, heads. So that's kind of an introduction to Punnett squares. What is the probability that an individual with genotype big A will give an allele big A to its offspring? So you guys, this individual is homozygous, right? It's homozygous A, right? A. Think of it this way. You know, it's big A on one side of the coin. It's big A on the other side of the coin. What's the probability that they're going to give a big A to their offspring? It's 100%. There's no chance of them giving a little a. They don't even have a little a, okay? Let's take a look at this guy. Let me get the eraser so you guys can read that. What's the probability that an individual with genotype big A, little a will give a big A allele to its offspring? So this is just like a coin with big A on one side and little a on the other. It's a 50-50 chance, a 50% chance, okay? List the probability of all genotypes for two parents with genotypes big A, little a, and big A, little a. So the way that this is worded, it's kind of poorly worded. It's supposed to say list the probability of all genotypes of the offspring. It didn't say offspring. For two parents with genotypes big A, little a, big A, little a. So I have on the next slide a Punnett square. Okay, so the parents are big A, little a. So for this parent here, big A, little a, it can give a big A or it could give a little a. Okay, so that's the parent here at the top. The other parent we're going to put on the side, that parent can give either a big A or a little a. So that's the whole point of a Punnett square. It kind of shows you like one parent is on the top, the other parent is on the side, the probability of donating a big A or a little a. Okay, and when you fill out a Punnett square, the big A, so let me just show you how to fill out a Punnett square. This big A is on top of this column, so you're going to bring it down. You're going to bring it down. This little a is on top of this column, so you're going to bring it down. And then this big A is in front of this row, so you're going to bring it across. So do you guys see now that I'm like filling in these boxes? And you can see that these are genotypes. Okay, these are genotypes. The two genes that they inherited from their parents. So it's asking like, what is the probability of like big A, big A, big A, little a, big A, little a, little a, little a? This one is 1 out of 4 or 25% big A, big A. Down here is also 1 out of 4 or 25% little a, little a. This other one, the heterozygous, let's start using the vocabulary here, okay? The heterozygous individuals, it's 1/4 and 1/4, which is 2/4, which is 1/2 or 50%. Okay, so 50% of the offspring, the probability is of these parents, so these parents, big A, big A, little a, big A, little, the probability of them having offspring that's big A, little a is 50%. The probability of them having offspring that's big A is 25%. The probability of them having offspring little a is 25%. So that's the whole point of Punnett squares, is that it's telling you the probability of what their offspring are going to be. Probability. Okay? Let's say that these parents, they have 4 kids that are little a, little a. And let's say that the mom is pregnant again with the 5th child. What's the probability of them having little a, little a? Some people would say, oh, they've had 4 kids with with 4 kids with little a, little a. There's no way they're going to have another kid. It's probably going to be big A, That's not how genetics works. It's all about probability. Every time they have a kid, it's 25% chance, little a, and just by coincidence, they've had 4 kids that are little a, little a. Next kid, probability is the same, 25% or 1/4. Okay? Alrighty. So the reason why we do a Punnett square is because of Mendel's law of segregation. So I did mention how at the beginning I said that Mendel came up with 2 laws. This is the first law, the law of segregation. And it states that I gotta move my head out of the way, but the 2 alleles of a single trait will separate randomly, meaning that there is a 50% chance either allele will end up in either gamete. So like I was saying, like big A, little a. So let's just say that on this chromosome is the big A, on this chromosome it's the little a. There's a 50-50 chance that the big A is going to go into one gamete. The little a is going to go into the other gamete. So it's going to be a 50-50 chance. Okay? And you can see that here. These guys have the big A. These guys have the little a. So you guys, this is, what does this look like? This is meiosis, right? This is meiosis. Remember in metaphase 1 of meiosis, the pairs, the homologous pairs from the parents, they line up at the metaphase plate. And then in anaphase and telophase 1 and cytokinesis 1, the homologous pairs separate. And then in meiosis 2, the sister chromatids separate. Okay? So there's a 50-50 chance of either giving a big A or a little a, because the law of segregation states the 2 alleles that you inherit from your parents, they're going to segregate. They're going to separate. There's a 50-50 chance of you giving the gene allele you got from mom. There's a 50-50 chance that you're going to give away to your kids the allele that you got from your dad. Okay? So that's the law of segregation. So Monohybrid crosses refer to Punnett squares that we're going to be dealing with one trait at a time. So mono means one, right? And we're talking about one trait at a time. So we're going to be dealing with Punnett squares, dealing with one trait at a time. So Punnett squares. Okay. We're going to be looking at flower color, seed color, texture of the seeds, like round versus wrinkled, the color of the pods, green versus yellow, the shape of the pods, inflated versus constricted, axial flowers versus terminal, where is the flower located on the stem, and then the length of the stem, tall stem, short stem, okay, and the numbers that are found in this table, and I believe this table is in your textbook as well. These are literally the number of plants that he counted, or maybe flowers that he counted. So remember how I said, like, Mendel took 12 years to study sweet pea plants, over 10,000 plants. Look at the numbers. I mean, when I do experiments, am I really going to count? So like yellow seeds versus green seeds. Imagine if your teacher, your science teacher, me, said, all right, guys, count how many offspring have yellow seeds. He counted 6,022 yellow seeds. He counted 2,001 green seeds. And the ratio of the F2 generation is the 3:1, 3.01 to 1. So he was looking at the F2 generation, the F2 generation. So when we say F2, we're talking about how you have the P generation and then you have the F1. F2 is the offspring of the F1. So you can see why it took so long to do this experiment, because he had it was like a generational study, okay, the F2 generation. My goodness, can you imagine having your teacher telling you to count this many seeds and pods? I think by the time you guys get to like 200, you're like, I give up. Let's just make up the numbers. That's not how science works, right? With science, you need to be accurate. You need to do a good job of counting. And that's what he did. Mendel loved math. He loved math and he loved botany. So really studying genetics for him with sweet pea plants was perfect. And we've learned a lot from him. So again, phenotype is the physical appearance. Genotype are the two genes that an individual inherits from their parents. Two organisms can have the same phenotype, the physical appearance, but have different genotypes. Okay? For example, if we have two purple pea plants, they have the same phenotype, but they might have different genotypes. How is that possible? So think about it for a second. And I'm lagging, I'm looking at myself. That's okay. So maybe the purple plant could be homozygous purple or heterozygous purple. Because remember, purple is dominant to white. So you're not going to see, in the physical appearance, you're not going to see the white gene. You're only going to see the purple gene. Okay? So how do we know, when we look at these purple plants, how can we determine the genotype? How do we determine, is it big P, or is it big P, little p? How do we do that? How do you determine the genotype of an individual with the dominant phenotype? There is something called a test cross, okay? A test cross. A test cross is where you breed the dominant phenotype with the homozygous recessive to determine the genotype. So here you have a purple dominant phenotype. Is it big P or big P, little p? You're going to do a test cross. Like cross means pollination. Test cross with a little p. Okay? This is going to tell you the genotype. It's going to tell you the genotype of this flower with the dominant phenotype. And this is how it works. How does test cross work? Well, if it was big P, if it was big P, what is the offspring going to look like? I'm going to go ahead and bring the big P's across. I'm going to go ahead and bring these little P's down. Okay? What are the offspring? 100% purple. Okay, now for the next one. Okay, that all just pops up. But for the next one, if it's heterozygous, I gotta move my head again. You know, for the next one, if it's heterozygous, so here's the heterozygous genotype. I'm gonna bring the big P across. I'm gonna bring the little p across. And then I'm gonna bring these I'm gonna bring these little p's down. And what do I get? 50% purple, 50% white. So do you guys see how a test cross, so I'm gonna write purple and then 50% white. So do you guys see how a test cross can tell you what is the genotype of the purple flower? If you cross it with the homozygous recessive, if you cross it with the white one, looking at the offspring will tell you exactly what the genotype of that purple flower is. If 100% of the offspring are purple, then you know it's homozygous purple. If it's 50% purple offspring, then you know it's heterozygous purple. Okay? Okay, so here are some sample monohybrid genetics problems. Let me see if I can. Oh, I wish there was some space on this slide so I can like write all over it, but it's not. There's not a lot of space. I'm just gonna have to do with. Okay. Some sample monohybrid genetics problem. Again, it's called monohybrid because we're dealing with one trait at a time. Okay. This is a long video already, and I'm not even like halfway done. So I'm drinking a lot of water. Okay, hypothetically speaking, brown eyes, big B, is dominant over blue eyes, little b. Just so you guys know, human eye color is much more complicated. It's not this straightforward, okay? So that's why I say hypothetically speaking. The gene is autosomal, not sex-linked. Do you guys remember when we talked about karyotypes? When we looked at the first 22 pairs of the karyotype, they're autosomal, and then the last chromosomes are sex chromosomes. So we're just saying that the brown-eyed and blue-eyed genes are not on the sex chromosomes. They're on one of the first 22 chromosomes, okay? All right, so for problem A, a heterozygous brown-eyed man. So what is he? It says heterozygous brown-eyed. So he has two different, hetero means two different. He's got brown eyes. So he must be, what's his genotype? He must be big B, little b. Heterozygous means two different. That's his genotype. He marries a blue-eyed woman. Blue eyes, right? Blue eyes is recessive, little b. So she must be little b, right? If she was big B, little b, she would not have brown eye, or sorry, she wouldn't have blue eyes because brown, big B, is dominant to little b. What will be the genotypes and phenotypes of the offspring? You know, it says what will be, but it really should say, what's the probability? What are the different probabilities of the genotypes and phenotypes of their offspring? Because there's space down here, I'll just go ahead and do the genotype here, the Punnett square here. So let's put the father here and the mommy here. So when I do the, oops, a daisy, oh, I think I can work with it. Yeah, little b, and then big B, little b, little b, little b. Okay, so I see 1/2 brown, 1/2 blue. So it says what will be the genotypes? It's going to be 50% or 1/2 big B, little b, 50% little b. And then what are the phenotypes of the offspring? 50% brown. So see how I'm like paying attention to the language here? Phenotype versus genotype. 50% blue. Okay, so I've just answered that first question, A, part A. And let's go ahead and answer B. The first child of the two brown-eyed parents. Hold on. The first child of the two brown-eyed parents has blue eyes. What are the genotypes of the three people? So the three people I'm talking about are the two parents and then their child. Okay, those are the three people that I'm talking about. What other phenotypes and in what proportion might be expected in future children? Okay, so the two parents have brown eyes. So we know that they must have a big B, because big B for brown. But then one of their kids has blue eyes. The first child has blue eyes. What must be their child? Their child is little b, huh? Okay, little b. So how is that possible? The parents have to be heterozygous. The parents have to be heterozygous to have a blue-eyed child right there. Okay, so let me go ahead and fill in the Punnett square. Because the question also asks, oh wait, what are the genotypes of the three people? Child is little b. Parents are little b, big B, little b, big B, little b. Okay? What other phenotypes and what proportion might be expected in future children? I see, it's asking about phenotypes. So how many are going to have brown eyes? 3/4, 3 out of 4, or 75%? So 3 out of 4 or 75% is going to have brown eyes. So listen to how I was paying attention to the vocabulary. It was asking me about the phenotype. It didn't ask me about the genotypes, because if it did ask me about the genotypes, and I would have to give a different answer, I would have to say 1/4 big B, 2/4 or 1/2 big B, little b, like that. Okay? Next question. There's a lot of, that's why this video is going to be long, because there's a lot of like sample problems, which I think that you might in the long run appreciate, because then it's going to help you, study for our quiz and also study for the AP exam. Like I said, there's going to be definitely genetics on the AP exam where you're going to need to know how to do these type of problems actually very fast, right? Because it's timed. Let me move this out of the way. Cuddles. A male mouse with brown coat was mated with Snow White, an alluring albino mouse. Ooh, okay. The resulting litter of seven young all had brown fur. The next time around, Snow White mated with Fluffy, who was the same color as Cuddles, but some of Snow White's litter were white. Explain. Okay, so Snow White is white. Okay, Snow White is white, all right? And so when she's mated with Cuddles, who is brown, they have 7 young, all brown. Okay, but then when she's mated with Fluffy, who is also brown, right? Who's also brown. So I'll write that down, brown. Some of Snow White's litter were white. Okay, so what do you guys think? Like, what's going on here? What would be the genotype of Snow? Well, we know what genotype Snow White is, because white is recessive. Does it say that here? It doesn't say that here, huh? Blah, blah, blah. Okay, but you know what? White, albino, that's a mutation. And most mutations are recessive. Okay, so Snow White. Let's say that's Snow White then. We can figure out what Snow White's genotype is. So we'll go with, since brown is dominant to white, we'll just say she's little b. Okay, and then big B is the gene for brown. Little b is the gene for white. Okay? Cuddles is brown. All of their kids had brown fur. What does that say about him? Right? What is his genotype? Most likely big B, right? Because if you were to do a Punnett square for Cuddles and Snow White, 100% of their offspring are going to be brown. So most likely Cuddles is homozygous brown, because all of his kids are going to be brown. Whereas when Snow White is mated with Fluffy, okay, and Fluffy is also brown, but some of the litter are white. Okay, so some of them are going to be white. So he must be big B, little b. Okay? I did this wrong. Sorry. It's like that. And then little b. Okay? So here are some of the white, and then here are the brown babies. So that means that Fluffy's genotype, oops, a daisy, that's supposed to be L. Fluffy is going to be big B, little b. There's this weird sound in my house. It's so bizarre. Okay, so Snow White is little b, Fluffy is big B, little b, and Cuddles is big B, big B. Next topic, dihybrid crosses, okay? And dihybrid, di means two, and we're talking about two different characters at the same time. So this is a study that Mendel did. Mendel, he studied not just one characteristic like flower color, pea color. He wasn't studying one thing at one time. He took it to the next level. He was studying like, I wonder if flower color affects pea color. I wonder if pea shape affects pea color. Like he was studying like how those genes might interact. And he discovered something. And he discovered the law of independent assortment. Okay, it comes from this whole idea of from his experiments of dihybrid crosses. So he was looking at two different characteristics at the same time, like seed color and seed shape. Those are two different characteristics that he studied in his experiment at one time. So this, I know this slide is very boring because there's so many words and no pictures, okay? But basically the law of independent assortment is a law that states that the alleles of two or more different genes get assorted into gametes independently of one another. In other words, the allele a gamete receives for one gene does not influence the allele received for another gene. And I'm going to show you guys a picture on the next slide. And we did talk about this in chapter 13 when we went over meiosis, okay? This law is only applied to dihybrid crosses where traits are found on different chromosomes. Okay, so here's a diagram. And when we talk about the law of independent assortment, we're really looking down here, okay? But let's start up here with the P generation. What did Mendel look at? In a dihybrid cross, he was looking at color. Oh, I don't know if you guys can see that with my screen recording. Because I noticed that with the screen recording, it cuts off the top and the bottom, okay? But he was looking at seed color and also the shape of the seed. Can you guys see that? I hope so. He was looking at seed color and shape. So with the P generation, he started off with purebred yellow. That's the color. So I'll write color here. And he was also looking at purebred round, which is the shape. So do you see how this is a dihybrid cross? Because we're dealing with two traits at the same time. Seed color, seed shape. So in the P generation, he started off with purebred yellow, purebred round. The other P plant that he cross-pollinated with was green, purebred green, which is recessive, purebred wrinkled, which is also recessive. So this is color, this is shape. Okay, and then when he cross-pollinated them, oh, I should say that the gametes that this big Y, big R individual can make is really only one kind of gamete, okay? Because it can only make a gamete with a big Y and a big R. It's sort of like, you know, like when you flip a coin, right? You have Y's on both sides. What are the chances of giving a Y? Big Y? 100%. What are the chances of giving a big R? 100%. So that's why in this picture right here, the gamete is a big R and a big Y. Okay? Same with this guy, 100% chance of giving a little y, 100% chance of giving a little r. That's why the gamete for this parent is little y, little r. Okay? And then fertilization takes place, cross-pollination. Mendel took his little paintbrush and cross-pollinated them. He ended up with yellow round seeds. Why was it yellow? Why was it round? Because remember, yellow is dominant to green, and then round is dominant to wrinkled. So they were all yellow and round, but they're all hybrids. So for the F generation, they're all hybrids. They're all heterozygotes or heterozygous for the trait. Okay? Okay, but getting to the law of independent assortment. You know, when the homologous pairs, when they line up at the metaphase plate in meiosis 1, they can arrange themselves in this way, where it's the dominant traits are on the left side, and then the recessive traits are on the right side. Or you could have recessive trait on the left, dominant trait on the right, dominant trait on the left, recessive trait on the right. Okay, do you guys see how there's like two different possibilities of arrangement? So if the two genes, so going back to the law of principle or law of independent assortment, if the genes for seed color, which is the Y's, or the genes for the shape, like round versus wrinkle, if they're on different chromosomes, the way that they go into the gametes is completely independent of each other. Do you guys see that? So here you have the gene for shape, seed shape, the gene for seed color, they're found on different chromosomes. So the way they will move into the gametes is completely independent of each other because they're found on different chromosomes, okay? So you could have it like this or like this. So depending on like how they arrange themselves in metaphase 1 of meiosis, you're going to end up with 4 different possible gamete combinations. Okay, so going back to the parents are both big R, little r, big Y, little y. So that's the F1 generation. The F1 generation, that's what they are: big R, little r, big Y, little y. And so the 4 different kinds of gametes that they can produce, there's 4 different kinds of gametes. Big R, big Y. Let's just draw it like sperm, okay? Or big R, little y. Or little r. This is going to get really messy, guys. Big Y. And then the last combination possibility is little r, little y. Okay, that little r and that little y. So there's 4 different possibilities of forming 4 different kinds of gametes here, okay? So that's the law of independent assortment going back to metaphase 1, but then also the law of segregation has to do with the homologous pairs separating, right? So do you guys see how, see how the Mendel's 2 laws, number 1 was the law of ooh, that is so messy. The law of segregation, and then number 2, the law of independent assortment, okay? And by the way, like, it's amazing because Gregor Mendel, he lived and he did his experiment back like in the 1840s or something. And then when was DNA, the structure of even DNA discovered? That was back in like 1950s, okay? So he was way ahead of his time. He had no concept of chromosomes and DNA. He just knew like these genes or these, he called them factors. These factors, they separate into the gametes independently of each other. I mean, amazing how he was such a smart guy, okay? So the law of independent assortment, which stage of meiosis creates the law of independent assortment? Metaphase 1. Remember the way that they arrange themselves? And metaphase 1 is completely random. The gene for maybe, seed color is here. So color gene is there. The gene for seed shape is there. The way that they line up is completely independent of each other. So exception, if genes are on the same chromosome and close together, it will usually be inherited together, okay? For example, with humans, I don't know which chromosome it's on, okay? But there's the gene for red hair and the gene for freckles are on the same chromosome and they're really close together. So they usually, when crossing over happens, they're usually inherited together because they're so close together, okay? They're inherited together. So usually when somebody has red hair, they have freckles, okay? Because they're on the same chromosome. This squirrel outside is insane. It's like digging around and maybe it's really hungry. Oh, so we say that the gene for red hair and freckles, they're linked. We say they're linked, and we say they're linked because they are usually inherited together. They're found on the same chromosome. So if two genes are found on the same chromosome, we say that they're linked. And we're going to be talking about that in chapter 15. Looking at pea color and pea shape, yellow versus green. So yellow is dominant to green. So the gene allele, the allele symbol for yellow, right, is big Y or capital Y. And because green is recessive, we're taking the same letter, but we are taking the lowercase version of that letter. Same with round. Round is dominant, right? So we're going to take the capital first letter of the word round. That's going to represent the round gene. But wrinkled is recessive, and we're taking the same letter, but lowercase, okay? What is the genotype of a pea plant that is heterozygous for both pea color and shape? So this is just asking you to really understand the language here. Remember, genotype are the two genes for heterozygous for pea color and shape. Okay? So heterozygous, yellow and green, and then pea shape, round and wrinkled. So that's the genotype. So this is the answer for this question. This is the genotype, the two genes, okay, for color and shape. And it says heterozygous. What type of gametes, sex cells, could a heterozygous pea plant produce? And we sort of went over that on the last slide, but just to clarify, so this guy right here that is heterozygous for both traits, this pea plant can produce 4 different possibilities, okay? By the law of segregation, those two genes are going to separate, right? Big Y, little y, okay? And then by the law of independent assortment, the way that they line up at the metaphase plate, you're going to get maybe big Y and big R coming together, or big Y and little r coming together, little y and big R coming together, little y, little r coming together. So there are 4 different possibilities. So you know what? It's easy to draw them as sperm again. So big Y, little r, right? I forgot to do that. And then little y, big R. And then little y, little r. Okay? So those are the 4 different possibilities of the gametes that can be produced by an individual that's heterozygous in a dihybrid cross. Dihybrid cross because we're talking about 2 different traits, pea color, pea shape. Okay? Oh, okay, and there's another question here. Let me go my eraser. What would be the phenotypic ratios of the offspring? Okay, it's actually the answers on the next slide. But what would be the phenotypic ratios of the offspring if 2 heterozygotes are cross-pollinated? So we're asking, you know, if the parents were big Y, big R, little r, what would be their offspring? And that sounds really complicated, right? Because there's 4 different gametes that each parent can produce. So you guys, this is not going to be a simple Punnett square. It's going to be a crazy Punnett square. It's going to be a 4x4, not a 2x2, because each parent can produce 4 different kinds of gametes. Okay, and so here they are. Right? So how do we fill out this Punnett square? You're going to bring the big Y's together. You're going to bring the big R's together. Big Y, big R, Y, big R, little r. So let me let the PowerPoint actually fill it in for us. Okay? So that's what you're going to end up with. Now, the question was asking about the phenotypic ratio, like the physical appearance. You know what? I do see, so whatever box I see that has a big Y, that's going to be yellow. So I'm going to write yellow. And then in terms of shape, I do see round. So I'm going to write yellow and round. You know what? I think that in the PowerPoint this all comes up anyway, so let me go ahead and just press the button. Okay, yeah, this one is actually physically showing you the phenotype. Okay. So yeah, some are going to be round and yellow, or yellow and round. Some are going to be yellow and wrinkled, and then some of them are going to be round and green, and then wrinkled and green. Okay. And if you were to count them, this is what you would get. 9 out of 16 yellow round. Actually, let's count them. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. Those are going to be yellow and round. 3 out of 16 are going to be green and round. 3 out of 16 are going to be yellow and wrinkled. And then the last guy right over here is going to be green and wrinkled. Okay? So this is what you would do for a dihybrid cross in terms of the Punnett square. But guess what? There's a way, way easier way to do this. You know, like on the AP exam, you don't have time to draw this massive dihybrid cross, 4x4, 16 square Punnett square, okay? There's a lot easier way. So let me actually find a blank, maybe Word document, and I'm going to have to, oh wait, oh no, what happened there? Okay. You know what? I might be able to just write it in the margins here. Okay, let's do that instead. Oh, but you know what? Then I don't have the pen option. I don't think I have the pen option. No, I lost it. Okay. I wonder what I should do here. Because if I was in Zoom, then I would have the Zoom pen option. You guys, I'm like technologically... But you know what? Let's just put it back up. And then I'll just draw here. Okay, let me see. Yeah, I'll just draw this way. This is good. Okay. So instead of doing a whole massive 4x4, what you can do is just separate the Y's from the R's. Hold on, I'll get a pen. Okay, so we could just do a simple Punnett square. Y and like that. So you're going to get big Y, big Y. Ooh, that doesn't look good. Big Y, big Y, big Y, little y, big Y, little y, little y, little y. So here I can see that it's 3/4. So 3/4 is going to be yellow, right? And then 1/4 is going to be green. And then let's do big R, little r, big R, big R, big R, little r, big R, little r, little r, little r. So here it's 3/4 round, 1/4 wrinkled. Okay? All right, so what conclusion can we come to? Well, you're going to end up with yellow and round. You're going to end up with offspring that are yellow and wrinkled. You're going to end up with offspring that are green and round and green and wrinkled. So let's do yellow round first. Let's do yellow round. So how many is going to be yellow and round? All you have to do, you guys, is you just need to multiply the fractions together. Yellow is 3/4. Round is 3/4. And you guys know how to do multiply fractions. You just multiply across, right? 3 times 3 is 9. 4 times 4 is 16. So 9/16 is yellow and round. Okay? All right, let's do yellow and wrinkled. Yellow is 3/4. Wrinkled is 1/4 or 1/4. And that gives me 3 times 1 is 3. 4 times 4 is 16. Now let's do green. Green and round. That gives me 1/4 times round, which is 3/4 or 3/4. So 1 times 3 is 3. 4 times 4 is 16. I feel like I'm totally, my computer's like totally lagging. This is weird. Okay, green and wrinkled. Maybe I'm running out of like memory space. I hope not. Green is going to be 1/4 times wrinkled, which is also 1/4. That gives me 1/16. Okay, and that gives me the same answers as if I did that massive, 4x4, 16 square Punnett square. If you can just do, in a dihybrid class, if you could just separate the the traits, the Y's from the R's, and just do 2 Punnett squares, and then do it with fractions. 3 out of 4 is going to be yellow. 1 out of 4 is going to be green. And then you just multiply them. You're going to get the same answers. Okay? So if I just move forward, you guys will see I'm going to get the same answers. And oh, whoa, whoa, whoa. Yeah, I'm going to get the same answers. 9/16, yellow round. 3/16, green round. 3/16, yellow wrinkled. 1 out of 16, green wrinkled. Okay? Okay, let's see. Let me see what time it is. I don't know how long I've been talking for, but it's been over an hour. Okay, what would be the phenotypic ratio of offspring if a pea plant that is heterozygous for both traits is cross-pollinated with a pea plant that's homozygous yellow, heterozygous round? So you guys, one of the things about these genetics problems, you really need to understand the language, okay? So the first plant is heterozygous for both traits, okay? That means it's going to be big Y, little y, it's going to be yellow, and then it's going to be big R, little r, it's going to be round, heterozygous, okay? With a pea plant that is homozygous yellow, so it's going to be cross-pollinated, homozygous yellow. Well, what's the genotype of homozygous yellow? Homozygous means 2 of the same, so big Y, because it's yellow. Heterozygous round, big R, little r. All right, guys, do we really want to do a massive dihybrid cross? Figure out like what kind of gametes that they can produce? Don't waste your time, right? Let's just do, you know what? I should probably show you how to do it that way first. Yeah, you know what? I'll show you how to do it that way first. That way you can see which way is easier. Or you know what? Maybe we're wasting time then, huh? That's okay. I'll just do it really quick. Oh, and then you guys could always fast forward, right? So, I mean, I'm saying like, oh, I feel bad for you, but at the same time, you're probably fast forwarding through this anyway. So, okay. So this guy, this parent, can make 4 different kinds of gametes, right? And then this guy can make how many different kinds of gametes? That's actually not 4, it's actually 2. Big Y, big R, big Y, little r. So this Punnett square is not going to be as big. It's going to be a 2x4. It could be a 4x4 if we doubled it, but we're not going to waste time doing that. So you could do it this way. Big Y, big Y, big R, big R, big Y, big Y. Oh, big R, little. Oh, it's so messy. Big Y. Now it's so quiet. That's the thing with these Punnett squares. They can take a while to fill out. Okay. All right. So let's figure out how many is going to be yellow and round. Yellow and round. So this one's yellow round. 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 This one's not yellow round. This one's yellow round. So I count 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. 6 out of, how many boxes are there? 8. 6 out of 8. We could probably reduce that, right? To 3/4, right? Okay, and then how many is going to be yellow and wrinkle, right? Wrinkled. Sorry, I forgot for a second what the little r. And I see 2. So this one. So 2 out of 8, and we're going to reduce that to 1/4. Okay, so that's the answer for that. That wasn't too bad, but let me show you the other way. The only problem with like doing it this way is you have to figure out what kind of gametes do they each produce, and that can be a little bit confusing. But if you just do two simple Punnett squares, you don't have to deal with the gamete situation, like what combinations of Y's and R's and the gametes are going to be made. You don't have to deal with that. Okay, so let's do the Y's first. And I'm keeping the answers up because we're going to end up with the same answers, and then the R's. So the Y's first, and then the R's. Big Y, big Y, big Y, little y, big Y, big Y, big Y, little y. Big R, big R, big R, little r, big R, little r, little r, little r. Okay, so you know what? 100% yellow, right? So I'm just gonna say that it's 4 out of 4. Okay, maybe just say 4 out of 4 yellow. And then over here, it's gonna be 3/4 round, and then 1/4 wrinkled. Wrinkled. Okay, so you know what? At 4 out of 4 is just 1. Duh, mission. Okay, so how many is yellow and round? So yellow and round is 1, right? That's yellow times round. 1 times 3/4 gives us 3/4. Isn't that what we got? We did. Our answers match up. Okay, and then yellow and wrinkled. I see yellow and wrinkled. It's going to be 1 times 1/4 for wrinkled. 1/4. Answers match up. Okay, wasn't that a lot easier? That was a lot easier, right? Than doing the whole like big Punnett square. What is the probability that two pea plants with genotypes big Y, little y, big R, little r will produce offspring with the genotype big Y, little y, big R, little r? Okay, so the parents are big Y, little y, big R, little r, and then they're asking, what's the probability that they're going to have a kid with big Y, little y, big R, little r? We could go back to that big, Punnett square and see the ones that are big Y, little y, big R, little r, but we don't need to do that. We could just do the 2 Punnett squares. Oh my goodness. This is taking a long time, this video. It's okay. I'm just trying to like fit the entire chapter into one video. Okay, and then so let's look at the first one. How many is big Y, little y? These 2 out of 4, right? See these 2 out of 4? So it's 2 out of 4, which is basically 1/2. Okay, and then how many of these is big R, little r? 1/2, right? So 1/2. So it's going to be 1/4. Okay, so the probability of them having a kid, big Y, big R, little r, is going to be 1/4. And if I go back to that big Punnett square, I believe it's like 4 out of 16. I think it's 4 out of 16. That is big Y, big R, little r. And 4 out of 16, if you reduce it, is 1/4. Okay. What is the probability that 2 pea plants with genotypes big Y, little y, big R, little r will produce offspring with a dominant phenotype? Okay. I am not going to redo the Punnett squares, but take a look at the ones above. Which ones have a dominant phenotype? If I look here, it's 3 out of 4, right? 3 out of 4 show the dominant phenotype. 3 out of the 4 have a big Y, so they have the dominant phenotype. So it's going to be 3/4. What about the R's? Also 3/4, right? Because I only see one that's little r. So these 3 out of 4 are going to have the dominant phenotype. It's 3 times 3 is 9. 4 times 4 is 16. So the answer is 9/16. That was pretty easy. You don't have to do a big Punnett square. What is the probability that the following parents would have offspring with dominant phenotype? You guys, now we're not dealing with dihybrid. What is this called? a quad hybrid because we're dealing with 4 traits. A trait, B trait, C trait, D trait. Are we gonna do a math? I think for this one it's like 120, I forget how many squares, but it's crazy, right? We don't need to do that. All we need to do is separate the A's from the B's, from the C's and the D's. Okay, so let's do that. A's, B's, C's, D's. Okay, so we don't need to do 100 plus Punnett square. Big A, little a. Big A, little a. So do you guys see that? I took the big A's, and then big B, little b. And then this one is little b, little b. And then this one is big C, and big C, little c. And then little d, big D, little d. Big A, little a, big A, little a, little a, little a, big B, little b, big B, little b, little b, little b, little b, little b, big C, big C, big C, big C, big C, little c, big C, little c. Big D, little d, big D, little d, little d, little d, little d, little d. Okay. Dominant phenotype. So we're looking for ones that have the big letter, the capital letter, the dominant phenotype. So here, 3 out of 4. Do you guys see that? 3 out of 4. Times. Which of these have a big B? 2 out of 4. 2 out of 4 is 1/2, right? C's. 1, 2, 3, 4. All. So 4 out of 4, which is just 1. And then big D's. 2 out of 4, which is 1/2. Do the math. 3. 4 times 2 is 8. Times 2 is 16. Bam. You got your answer right there. Okay. So, did I just say bam? That's so cheesy. 4 times 2 is 8. Times 2 is 16. So the answer is 316. 316 chance that they're going to have an offspring with a dominant phenotype. You do not need to do the 100 plus Punnett square. Oh, okay. Next question is down here. What is the probability that they will have offspring with recessive phenotype? Dang it. Let me move that out of the way so I can actually see. So 1 out of 4 times 1/2. Okay, here's the dealio, guys. Do you see any with the recessive phenotype? 0, none. And then guess what? You can stop your math there. Don't waste your time, right? Because anytime you have a 0, you multiply it by 0. What's the answer? 0. Okay? You guys, I think I'm going crazy because I've been talking for so long. Now, you guys, we're going to talk about non-Mendelian genetics, okay? Non-Mendelian genetics means that we're not talking about complete dominance. What we've been talking about is complete dominance. Like purple is completely dominant over white. Yellow is completely dominant over green. Incomplete dominance, think of it this way. Incomplete dominance is going to give you a color that's supposed to be an equal sign. In between. That's how I remember this. Incomplete, in between. So like if you mix red paint, mix it with white paint, what color do you get? An in-between color, pink. Pink is in between, okay? So here we're gonna use different letters because this is incomplete dominance. We use capital, lowercase letters when we're talking about complete dominance. But when it comes to incomplete dominance, we use 2 different letters to let us know that this is not a Mendelian genetic problem. This is not a complete dominant problem, okay? So for red flowers, you need 2 red genes. Big R, big R. 2 red genes to give you red. You need 2 white genes to give you white. And then you need red and white to give you a pink phenotype, okay? So you guys, these are the phenotypes. These are the genotypes. Do you see that? Phenotypes versus genotype. So there were other scientists, I don't think it was Mendel. He didn't study incomplete dominance. That's why it's called non-Mendelian genetics too, okay? So with non-Mendelian genetics, these are called snapdragon plants, and snapdragons show incomplete dominance. My camera is like super duper weird. It's like my face is like blown out, but it's okay, whatever. Let me click here. So you have true breeding red. That means it's big R, big R. True breeding white, that means it's WW. If you cross-pollinate them, they all are the same. They all get a red gene. They all get a white gene, right? So they're all big R. Oops, a daisy. That's supposed to be big R, big W. I was going to write something else there. Let me get my eraser. Yep. Okay, so it's supposed to be big R, big W, okay? They're heterozygous, they're hybrids, they're 100% pink. When they self-pollinate, let me do a Punnett square. When they self-pollinate, big RW, big RW. You get 25% red, 50% pink, 25% white. Do you guys see that? 25% red, 50% pink, 25% white. So that's the F2 generation. Okay, that's the F2. So the parental generation, P generation are true breeding. F1 generation are hybrids, and then the F2 generation, you see this 25 to 50% to 25% ratio. Okay. All right, here we go with another practice problem. Crosses between yellow rat and a yellow rat produces yellow rats. Crosses between a white rat and a white rat always produces white rats. But crosses between a white and a yellow rat produces a cream rat. So think of it this way. Yellow and white, when you mix those two colors together, you get cream. So the cream color is the incomplete color. It's the mixing color. It's the in-between color. It's the heterozygous color. What are the genotypes and phenotype ratios if you cross two cream rats? Okay, so maybe it's better to just do it this way. Big Y is yellow, and then W is white, right? So like this would be a yellow rat, and then this would be a white rat. But then a cream rat would be that one. Okay, and it's asking cross two creams. So we're gonna draw. So the beauty of like doing a recording is that you guys could press pause and then work on this on your own, and then you can fast forward, okay? What is the genotype and phenotype ratios if you cross two creams? So it's gonna be 25% big Y, 50% big Y, little y, 25%, WW, what the heck? I'm not saying it right. 25% YY, 50% YW, 25% WW, or 25% yellow phenotype, 50% cream, 25% white. Okay? Do you guys find this to be fun? I find this to be interesting and fun. I hope you guys are too. Oh my god, this is like so much. You know what we could do is you guys could do this on your own, and then we can go over in class. But it's basically incomplete dominance, okay? If you think that two alleles for coat color in mice show incomplete dominance, what is the best? Oh, I see. This is actually not a math problem, okay? What is the best and simplest cross to perform in order to support your hypothesis? So you think that this coat color shows incomplete dominance. Which of these, A, B, C, D, or E, can give you the answer to that question or to verify that hypothesis? A test cross of a homozygous recessive mouse with a mouse of unknown genotype. Okay? B, a cross of F1 mice to look for a 1 to 2 to 1 ratio in the offspring. So you guys remember with the F1, so let's see, you have yellow, that's yellow, right? Oh, I should probably write, yeah, like this. And then this is going to give you white, and then this is going to give you cream. Okay, so remember in the P generation, that's the P generation, they're purebred, and then the F1, that's the F1, they're like hybrids. And what this is saying is that have the F1 generation breed, and then you should see them come up with this 1 to 2 to 1 ratio where it's going to be 1 yellow, 2 cream to 1 white. Like, let me go back to this one, this thing. It's saying like, if the F1 generation were to reproduce, then you would get this 1 to 2 to 1 ratio, 1 red to 2 pink to 1 white. And that's true, right? So the answer might be B. Let's go ahead and look at C. A reciprocal cross in which the sex of the mice of each coat color is reversed. What? The sex of the mice of each coat color is reversed. That doesn't even make any sense to me. D. A cross of two true breeding mice of different colors. A cross of two true breeding mice. So like yellow and white of different colors to look for an intermediate phenotype in the F1. Okay, that could be it too, right? Because if you take two true breeding mice, like white, and cross it with the yellow, that's going to give you a cream, an intermediate. So that's possible too. Okay, and then E, a cross of F1 mice to look for a 9:7 ratio in the offspring. No, that's not it. So it could be B or D. So the question is, what is the best and simplest cross to perform in order to support your hypothesis? So the simplest one would just be to take two true breeding mice, cross them, and then see if their kids have that in between. In between is incomplete, the intermediate phenotype. So I would say D is the simplest way. B is an alternative, but it's not the most simple way. Okay? All right, next problem is codominance. You know what? I'm just going to take a quick break here, and then I'm going to come back. But it doesn't matter to you because video just continues on. But I'm going to take a really quick break. Okay? Back. I just took a little 2-minute break, and we are about halfway through. But I think we can get through this second half a lot faster than we have been going. I hope. All right, the next topic is codominance. Noah, sorry, my son is here and I'm going to ask him to leave. Can you leave, please? Thank you.
Hello. Bye now.
Oh, yeah. So that first part was like an hour and 31 minutes, an hour and 31 minutes long. And he was like, when are you going to come out and play? And I'm like, oh. All right, so codominance is similar to incomplete dominance, but it's a little bit different because if you think about the word co means together. So in this case, you're not getting an in-between color, but you are seeing the two colors show up together. So in this case, the two genes are showing up together. They're not combining into one to give you an incomplete phenotype, but you're seeing both of them independently showing up together. So for example, human blood groups, okay? Human blood groups, you have A blood, you have B blood, you have O blood, you have AB blood. You know, there's multiple alleles. This is an example. Blood type is an example of not only codominance, but blood type is also an example of multiple alleles, meaning that there's more than two alleles for a trait. You know, like with the pea color, there was like always two genes, right? Purple versus white, or green versus yellow. And then even with the rats that we were just talking about, white versus yellow, those were the two genes. But with blood typing in human population, there's 3 main blood types: type A, type B, type O blood genes, okay? And A type blood gene and B type blood gene are codominant, meaning that if you have a heterozygous person that inherits both genes, they're going to show up at the same time. Whereas the O blood type is recessive. So these are the symbols for blood types: IA, IB, little I. So it's little I because little I is recessive, whereas the capital I's, IA, IB, are going to be codominant. I'm so lagging. My audio will not match up with the video. And that is very annoying. All right, this is what a phenotype looks like. Okay, the phenotype looks like. So if you have type A blood, your red blood cells have these proteins on the outside. You know what? I just realized it sort of looks like coronavirus, but there are these glycoprotein. In this case, it's actually carbohydrates, okay, and proteins together. But with coronavirus, it's actually proteins that are sticking out. But here, you're going to have carbohydrates on the outside that resemble the group type A gene. Okay? But if you have the IB blood type, so this is going to be IA. IB blood type, then this is what the carbohydrates look like on the outside. But then if you have both genes, they're co-dominant. Okay, you're going to see both carbohydrates on the outside. And then if you have O blood type, little I, there are no carbohydrates on the outside. So the carbohydrates are like referring to the tips right here. Okay, so if you have A blood type, you could either be IA, or what's the other what's the other genotype? You could be IA little I, because remember, little I is the O blood gene, which is recessive. And then if you have B blood type, you could be IB. I'm running out of space here. Or you could be IB little I, right? Okay. Also, just something that's totally, we need to get through this PowerPoint fast, but you also have antibodies in your blood. And these antibodies are basically produced by your your B blood cells that are looking out for the opposite blood type. So anti-B antibodies, it's looking out for, if you have A type blood, it's looking out for these guys, the B blood. It's going to attack the B blood red blood cells. Okay. If you have B blood type, you have anti-A antibodies. They're going to attack the A type red blood cells. And then if you have AB blood type, you don't have any antibodies because you show both. So you don't want to make antibodies that are going to attack either the A or the B, right? And then if you have O blood type, you have both antibodies that are going to attack the A type and the B type. Okay, so people with O blood type, because they don't have the carbohydrates on the outside, they are the universal donor. Anyone can take their blood because there are no carbohydrates to be recognized by the antibodies. So if you are O blood type, you should be the donor. And then if you have AB blood type, which is the rarest type of blood, you're the universal recipient. Because you don't have any antibodies that are going to attack any blood type, you can receive any type of blood. Okay? All right. That's why this PowerPoint is so long. This video is so long because of these practice problems, and like my back is hurting now, my neck is hurting. The father of a child has type AB blood. Okay, so what's his genotype? That means the father is IA, IB. The mother has type A. Mother is IA. She could be IA, or she could be IA little I. Which blood types can their children not have? Okay, so it's basically asking, if we were to do a Punnett square, and we have to actually do 2 because there's 2 possibilities for her, right? Actually, let's put the guy on the side here. The guy on the side. You guys, I think I'm going crazy. I've been talking for so long. I think I am so tired. Oh, yeah, then it's a little I. Okay, so if I were to fill this in, which I'm not going to bother filling in, the question is which blood types can their children not have? You know what? I do not see little I, right? So they cannot have O blood type. Can they have A type? Yes. Can they have B type? Yes. They can have B type right over here. IB little I. That's a B type. They can have A type. And they can have AB type. This is the AB type right over here. AB type is going to be down here. So the answer is O blood type. They cannot have any kids with O type. Okay. Oh, okay. Let me erase this. I wish like in Zoom there was like a erase all button, but there is no erase unless yeah, I don't see it here. The mother has AB blood, the father has type B. I believe that's what it says behind my face. His mother has type O blood. What are the possible blood types for their children? Okay, so let me go ahead and I wonder why it's not erasing some of the lines. Alrighty. So the mother has AB blood type. I'm trying to get the pen to show up. Come on. Oh my gosh, this is... What is going on here? Okay, there we go. All right. AB for mom. Father has type B. I think that's what it says behind me. Let me just move my... Yeah, type B. His mother has type O. You know, his mother is little I, little I. So what does that mean that he is? He had to have inherited one of the O blood type genes. So he has to be IB because he's B blood type, little I. Remember, B blood type, A blood genes are dominant to O. Okay? What are the possible blood types for their children? So if you do the Punnett square, IA, IB, IB, little I. Okay, so I'm just going to write in the letters because just to simplify it. BB, AI, BI. Okay, so they could have a B blood type. There's a 25% chance, right, of AB blood type. There is a 25% chance. Oh, yeah, 25% chance of A blood type. And then there's a 50% chance of B blood type. These guys right here. Let me just double check my work. A, B, 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 A, B. Yeah. Okay. All right. This next one is kind of a controversial one. Well, not controversial, but drama, right? The mother has type B blood. Her husband has type AB blood. Their child has type O blood. The father claims that the child can't be his. Is he right? He's got O type blood. So what's his genotype? It's little I, right? If he's little I, oh, sorry. The kid is little I, sorry. The child is little I, right? The dad is AB blood. Can he be the dad? Can he be the dad of this kid? No. Okay, he can't be. So he is right. He claims that he cannot be the child's dad. Sad story. Okay. Oh yes, now we're gonna move on to other stuff. Pleiotropy. So pleiotropy, most genes are pleiotropic, meaning it's one gene affects more than one phenotypic characteristic. An example of this would be like, besides this example called achondroplasia or gigantism, let me just think. Like sickle cell anemia. Somebody with sickle cell anemia, their hemoglobin, there is a mutation in the hemoglobin, and because there's a mutation in the hemoglobin, it changes the shape of the red blood cells, which can have multiple effects like blood clotting, stunted growth, pain in the muscle, heart problems, etc. Like pleiotropy just means one gene can have a wide variety of effects, phenotypic effects, okay? So achondroplasia is a type of genetic condition of dwarfism, and as a result, you can have lots of different phenotypic characteristics besides short stature. You know, you can have some respiratory problems. You might have like your fingers are shorter, toes are short, things that have multiple effects, okay? Same with gigantism or acromegaly. Acromegaly. Oh, you know what? We didn't talk about the foxes. I'm sorry, Fillmore. Acromegaly was seen in Andre the Giant. So Andre the Giant, he was I guess a wrestler, and then also he was an actor as well. Like he was in The Princess Bride. Do you guys like The Princess Bride? That's an old classic. So he had acromegaly or gigantism. So achondroplasia is again a type of genetic condition of dwarfism, but it actually is a dominant trait, which is very rare. Like I was saying to you guys how with mutations, usually mutations, genetic disorders or mutations are recessive, but actually achondroplasia is a dominant trait. So the gene for achondroplasia is going to be big A. Achondroplasia. Okay? So if you get a big A, you're going to have dwarfism. So here we have an individual with dwarfism, and then you have an individual here that is of normal height, then there's a 50-50 chance. Okay? And then another thing is, like, if you have two people with dwarfism or achondroplasia, there is a 25% chance of them having an offspring that is going to be big A, but unfortunately that is a lethal combination, so that embryo or that zygote will never develop. So as a result, you end up with a Punnett square that's actually 3, okay, 3 squares. There is a 2/3, so 2/3 chance right here of having a child with achondroplasia, and then 1/3 chance of having an offspring of normal height. Okay, and then there's this, so that was pleiotropy, and then there's also epistasis. Epistasis is where one gene completely masks another gene. So one gene completely masks another gene. So for example, little c, that combination, that is recessive, right? It's homozygous recessive combination, but that's going to give you no color in the coat. No color in the coat of, like, these mice. So if these mice get little c, they're going to have no color, even though they may have inherited the black gene or the brown gene. So we don't have a whole lot of time to go through this, but if you look at this dihybrid cross, you're looking at black versus brown or pigment versus no pigment. So if you look over here, these mice have inherited the little c. That means that they're going to have no color. They're going to be white. They're going to have no color. But notice how there's a big B. So what color should it have been? If it didn't have the little c, it should have been black. This one should have been black. This one should have been black. And then this one should have been brown. But because it inherited the little c, there's no color. It will not show up the black. It will not show up brown. So this is epistasis, where one gene completely masks another gene. Okay, moving forward. Now we have polygenic inheritance. I don't know if you guys can see that. It says polygenic. Polygenic means many genes. So polygenic. So many, right? Many genes, which are going to lead, so that's supposed to be an arrow, it's just going to lead to one phenotype. You know how we said pleiotropy is where you have one gene, many effects? Well, this one is kind of the opposite. It's many gene, one phenotype, one effect, okay? So for example, skin color. Skin color is due to multiple genes. In this example, it's due to 3 sets of genes, A, B, C, okay? And the more dark genes you inherit out of the 6, the darker your skin color. So it's like an accumulation, it's an additive effect, an accumulation of effects. You add all of the dark genes together, is going to equal darker skin. If you have more lighter skin genes, then you're going to have lighter skin tone. If you have half and half, 3 dark, 3 light, then you're going to be right down in the middle. Okay? So many genes, that's going to give you one phenotype, like skin color. But you know what? Skin color is also epigenetic. Like if you go out to the sun and get a tan, right? If you expose yourself to heat, your skin cells or melanocytes will produce more melanin, okay? So even though I'm saying skin color is like many genes, one phenotype, that one phenotype is actually, there's a range, okay, within that one phenotype. Height as well, weight, intelligence, behaviors, those can be genetically determined by multiple genes, okay? Okay, next topic is sex-linked traits, which we're going to discuss in further detail in chapter 15, but these are basically genes that are found on the sex chromosomes. Remember, for humans, there's like XX for female, XY for male. Same with fruit flies. Fruit flies are also XX female, XY male. Okay, Drosophila is the scientific genus for fruit fly. Drosophila melanogaster is the genus species name for fruit fly. They're a really good genetic subject to study because they only live for like 2 weeks. They can reproduce very quickly. And also they only have 4 pairs of chromosomes. So their genome is very small. So they're heavily studied because it's easy. We mapped out their chromosomes. We know where all the genes are on each chromosome. We could do knockout gene studies where you can like cause a mutation and see what happens with that gene, okay? Or see what happens if we like cause a mutation. Okay, so remember we have the first 22 pairs are called the autosomes, okay, or the autosomal chromosome, and then the last 2 are the sex chromosomes. And you guys should know just by looking at this karyotype that this is a male, right? Sex chromosomes, X, Y. So there was this scientist named, last name was Morgan. So his last name was Morgan. I forget his first name. But he was the first to discover that in fruit flies, the gene that determined the eye color, like red color versus white eye color, is on the X chromosome. So he was the first to discover that eye color in fruit fly is linked to sex, okay? It's on the sex chromosome. So true breeding red-eye female crossed with true breeding white-eyed male, when they mate, they produce 100% of the offspring are going to have red eyes. And then he had the F1 generation, the hybrids mate, and he discovered that all the females had red eyes, but then half the males had white eyes. So he was wondering, like, every time he found a fruit fly with white eyes, why was it always a male? It was always a male. So he was the first guy to discover that the color for eye color is found on the sex chromosome. Okay, so let's talk about that. So I should probably write down the genotypes, okay? So the red color versus white color. White color is recessive to red. So red is going to be dominant to white. However, because it's found on the X chromosome, this is the way that we draw out or write out the genotype. She is homozygous red eye. Homozygous red eye. Okay? So this is the genotype of the true breeding red-eyed female. X, big R, red. X, big R, red. So it's found on the sex chromosome. Okay? Let's write out the genotype for the male. He is wide-eyed. So that's going to be X little r. Okay? So maybe I'll write that over here. X big R is for red eyes. X little r is for white eyes. And because he's a male, he does not have 2 X chromosomes. I always have students like writing, oh, he's a wide-eyed male, X little r, X little r. No, that's a wide-eyed female, okay? He has a Y chromosome. So that's his genotype, X little r, Y. So you know with Morgan, he discovered that whenever he found a wide-eyed fly, it tended to be a male. And the reason why is because males only have that 1 X chromosome. Whatever gene is found on that X chromosome is going to be expressed. And in this case, it happens to be white. Therefore, and then white is recessive, it's going to show up as having white eyes. Whereas a female, like this female right here, that female is a hybrid. It's a heterozygous red-eyed female. Why does it have red eyes even though it inherited white? Because red is dominant to white. So let's go ahead and do the Punnett square. So the mom, X big R, X big R. The dad, X little r, Y. Ooh, this is going to get super duper messy because the Punnett square is very small. Hopefully you guys can understand this, okay? So I see females, X big R, X little r, X big R, X little r. So this female right here, her genotype is X big R, X little r. And then the male right here is X big R Y. X big R Y. So all the males are the same. They're all X big R Y. Okay? So all the females are X big R, X little r. Red eyes, because again, red eyes is dominant to white and it happens to be sex linked. That's why it's on the X chromosome. The males are also red eyes. And if you think about it, like males, where do they get their Y chromosome from? Their dad. Their dad gave them the Y chromosome. Where do they get their X chromosome? From their mom. And their mom is X big R, right? So that big X big R came from the mom. Okay. And then in this experiment that Morgan did, he had them mate with each other, and then he found out that all the females had red eyes. So wish me luck on drawing the Punnett square for this one. So then we have the females. All the females are X big R, X little r. All the males are X big R, Y. So let's go ahead and fill in this Punnett square. See how all the females have red eyes? And then half the males have red eyes, half the males have white eyes. Do you guys see that? This is the red-eyed male, this is a white-eyed male. All the females have red eyes. So 100% red-eyed females, 50% red-eyed males, 50% white-eyed males. Okay? So this is how he determined by, like Mendel, he, cross-mated, and he found out that red is dominant to white, but then also the gene for red eyes and white eyes are found on the X chromosome. Hemophilia is carried on the X chromosome and is recessive. Cross of woman carrier for hemophilia to a hemophiliac man. Okay, so she's a carrier. Carrier means that she carries the recessive trait, but she does not show it phenotypically because it's a recessive trait, and she has a normal dominant trait to express, to show. Okay? And then he's hemophilia. So hemophilia is a genetic disorder that is recessive. So I'm going to write X big N for normal. X little n is going to be hemophilia. Sorry, I got distracted there. I heard some shouting. I don't know what's going on. So she's a carrier. That means she must be normal, but she carries the hemophilia gene. Do you see that? That's a carrier. That's a female carrier. And then hemophiliac man, he has hemophilia. So like I was saying, males only have that one X. Whatever they get on that X chromosome is what they're going to express. They're kind of just stuck with that X. Whatever X chromosome they get from their mom, they're stuck with, okay? So then we'll do a Punnett square. So let's just put her on the top here, and then the dad on the side here. X big N, X little n, X. By the way, I'm writing super messy, but when you guys do Punnett squares, I think it's really important for you to be neat, because then it won't be so confusing. Like, this might confuse you if you wrote as messy as I did. Okay, what fraction of the offspring will be carrier females? So it's asking about out of all the offspring. And here, maybe I'll use a different color. Just so I can, like, let's use blue, okay? So the carrier, I only see one carrier female, 1 out of 4. So I'm gonna say like 1 out of 4 is gonna be a carrier. What fraction will be normal males? I only see one, this guy, normal. The other guy is going to be a hemophilic, so I'm going to say 1/4. What fraction will be normal females? You know what? This female inherited 2 hemophilia genes, and so it's 1 out of 4. 1 out of 4 is going to be not have the disease. Okay? Oh dear. Okay. I need my eraser now. Oh, I hope you guys are hanging in there. I'm sure you guys are fine, right? I mean, you can pause, take a break whenever you want. It should be me that you feel sorry for. I'm just talking nonstop for like 2 hours. My goodness. And here's the thing, like I'm normally a very quiet, shy person in person. Like like in staff meetings, teacher meetings, I'm like super quiet. I never speak up. But with you guys, I like, you can't shut me up, right? Okay, the gene for color blindness is carried on the X chromosome and is recessive. So I'm going to write this like this. So the gene for normal color vision is going to be X big N. It's normal. And then color blindness is X little n, color blindness. The reason why I keep on choosing like N is because N is dominant, it's normal. So normal is dominant. Little n is going to be the genetic disorder or genetic condition that's recessive, okay? Let's see, a man whose father was colorblind has a colorblind daughter. Is this, sorry. All right, let me get a drink of water. Is this man colorblind? How do you know? So this is the man that we're talking about right here. His dad was colorblind. So let's figure this out. If his dad is colorblind, what's his genotype? It's going to be X little n Y. So this man has a daughter who's also colorblind. So what's the daughter's genotype? She's got to be X little n, X little n. So the question is this man colorblind? You know what? He has to be colorblind because look at the daughter. The daughter inherited 2 genes for colorblindness. One is from mom, one is from dad. So how do we know? Because when we look at the daughter, she had inherited 2 colorblindness genes. One from mom, one from her dad, the man. Okay? B, where did he get his gene for colorblindness? So he's colorblind, right? He's colorblind, X little n Y. Where did he get this gene from, everybody? And if you were here in class, you would be saying, his mom. He got it from his mom because the Y comes from the dad, okay? Must the fathers of all colorblind girls be colorblind? All colorblind girls are X little n, X little n. Yes, their dad must be colorblind because that X little n comes from the dad. He only has one type of X chromosome to give to his daughter. He must be colorblind. And then the other one comes from the mom. Okay? Whew. Okay. Gosh, I almost feel like maybe we should skip these in the video and then maybe go over them in class because it's already for me, it's 4:15. Let me, blah, blah, blah. Yeah, I think, yeah, these we definitely can go over in class. Maybe we can go over this one with the cats because it might intimidate you, okay? So maybe we'll go over this because this is also co-dominant. It's sex-linked and co-dominant, which is interesting. In alley cats, the code is determined by a gene carried on the X chromosome. At the same time, the alleles are expressed as co-dominant. Genotypes and colors are as follows. Females, X little b, yellow. Oh, that's interesting, you guys. This is co-dominant, but for this problem, they use capital and lowercase, and that shouldn't be true, right, you guys? It should be XY. X. Oh, maybe they didn't do that because it's too confusing, like a sex chromosome. That's maybe why. I'm going to go ahead and erase that. But you guys know what I'm talking about, right? With co-dominance, you're supposed to use 2 different letters to show that it's co-dominant. Because if it was a capital and lowercase, that means it's dominant recessive. Anyway. So this is what would give you yellow. This would give you, so heterozygous is going to give you calico. Hetero. And then black is X big B, X big B. Males, yellow, X little b, Y. Males, black, X big B, Y. So you're not going to find any calico males, so no calicos. And when I say calico, calico is like the tortoiseshell cats. They tend to have like yellow, orange, and black mixed in their fur. So whenever you guys see a cat that is orange or yellow and black, it's most likely female. Very rarely do you ever find males that are going to have the two colorations, okay? Calico cat. So calico cat, X big B, X little b, has a litter of 8 kittens, 1 yellow male, so 2 black males, 2 yellow females, and 3 calico females. What is the color of the father of the litter? Okay, so this is the mom. So I'm going to do a Punnett square. This is, let's put the mom on the side. X big B, X little b. It's asking, what's the dad? What's the dad? I mean, this is the litter here, right? So the dad has to have a Y. So that's easy, right? The dad has to have a Y. What's the X chromosome? So you guys, we can see here that we get, maybe we should just fill this in. Yeah, let's just fill this in. So I can see the males here, right? 1 yellow male. This is the yellow male. These are the 2 black males. But what about the females? 2 yellow females and then 3 calicos. So these are the females and then these are the calicos. So what must be the color of the dad? He's got to be, in order to get calico, we need the X little b, right? So he's got to be yellow. He's got to be yellow. Do you guys see that? Okay. In order to get a calico female, you need that yellow in there. And then also there's 2 yellow females, and that's these guys. So these are the 2 yellow females, and then these are the 3 calico females. Oh, okay. We're going to skip that. Oh my goodness. Okay, the last thing in this presentation is about pedigrees. So we're almost done. But there's a lot with the pedigrees. I think I'm going to take a little quick break, okay? And it doesn't matter to you guys because I'm just going to continue the video. But I do need a break. Okay. Anyways. Okay, so I'm back, but I just had a little snack too. These little cookies. Have you guys ever tried these before? It's the chocolate. Anyways, it's so good. Really good cookie. Because I need that sugar. I'm burning a lot of I'm using up a lot of ATP, so I need sugars to make more ATP and cellular respiration. Do you guys remember? Glycolysis, Krebs cycle, the ETC. Okay, anyway. So pedigree analysis. A pedigree is a diagram that shows you how a specific trait is inherited through several generations. Okay, so pedigree, you're going to see squares and circles. Squares represents males, and then circles represent females. And then if the square or the circle is filled in, that means that that individual in that pedigree, in that generation, has that trait. And the trait could refer to a genetic disorder. It could refer to a color eye. It could refer to, in this case, these 2 pedigrees, it's referring to having a widow's peak or having an attached earlobe. Yeah, so having a widow's peak or having an attached earlobe. Those are like the 2 traits that are being studied in these pedigrees. So widow's peak. I have a widow's peak. Do you guys see that? It goes down, okay, in the middle. It's so annoying. I don't really like it because anyway. So if you have a widow's peak, which is a dominant trait, it's going to be filled in. So here's a male who has the widow's peak, so it's filled in. It's a dominant trait, so he's heterozygous. He has a partner, a female who is little w, no widow's peak. And then this line means that they're connected, they have offspring, and they have how many offspring? 1, 2, 3, 4. Specifically, they have 1 daughter. So it's usually also by like age, like the daughter going from left to right. This is the first child is a daughter. Second child is a son. Another son. 4th child is a son. Daughter has the widow's peak. She's heterozygous. The two sons in the middle do not have a widow's peak. The last son does have a widow's peak. The last son is connected to this female who is also heterozygous for widow's peak. They have 2 daughters. The first daughter has the widow's peak. The second daughter does not have the widow's peak. And you guys can see here that the daughter that has the widow's peak, she could be homozygous, widow's peak, or heterozygous for widow's peak. If you did the Punnett square, you guys would see that there is a, a possibility. So maybe I'll just draw it. So they're both big W, little w, big W, little w. So you can see here, it could be big W, or big W, little w. Okay. So you guys, that's basically what a pedigree shows. It shows multi-generational, what am I trying to say? It shows the traits in a multi-general, oh my God, I think that snack has really messed up my ability to speak. Or maybe this is just the way that I am, right? So pedigree, it shows how a trait is passed down from generation to generation. Multi-generational, okay? And then also free earlobe. Free earlobe is a dominant trait. I was talking to one class about this. Like I have a free it's kind of hard. So I have a free earlobe. It's not attached to the side of my face. So this is what an attached earlobe looks like, free earlobe. But I do know what I am because in the attached earlobe is going to be colored in. My mom, so let me just draw my pedigree. So my mom has an attached earlobe. So she's obviously little f. But my dad, he had a free earlobe. And I don't know if he was homozygous or heterozygous, but I'm their daughter. Oh, yeah. And I'm sorry, I was going to draw a square for a mistake, and that's male. I have a free earlobe, so I must be big F, but then I know what my mom is because she has attached earlobes, so she must have given me the little f. So I know for certain that I'm heterozygous for free earlobe. And these pedigrees are often done, a family history is often taken when dealing with genetic disorders. So if you're going to be a genetic counselor, oftentimes you have to look at a family tree to figure out if a trait can be passed down to your kid, to the couple's kids. So if you're a genetic counselor, you would meet with couples and you discuss, like, the possibility of passing down a specific genetic trait. So here's a simple pedigree analysis. What type of inheritance pattern would this pedigree indicate? And when we say inheritance pattern, the question is, number one, is this autosomal or sex-linked? So that's the first question that we need to answer. And then when we also say inheritance pattern, we have to figure out, number two, is this dominant or recessive? So I'm going to change the color because it's the same. Number two, is this dominant? Oops, dominant or recessive? So when we do these type of pedigree problems, the first thing you have to figure out is this autosomal or sex-linked? Is this dominant or recessive? Okay. So the two parents here do not have the condition. They have 4 children. Their eldest son has the condition. Here we can't really tell that that it's sex-linked. Okay. We can't really tell that it's sex-linked because the son is going to get the Y from the dad. And when we say sex-linked, it's usually the X chromosome we're talking about. He got the Y from his dad. The mom has 2 X chromosomes. This condition that we're talking about, it could be either or. Okay, so we can't really tell if it's sex-linked, but we can tell if it's recessive or dominant. Okay, we can't tell if it's recessive or dominant. So how can we tell that this is recessive or dominant? The two parents do not have the condition, but one of their kids does. That means this is recessive. That means that this is recessive because the parents, maybe it's like big A, little a, big A, little a. So they have the dominant condition, right? And then if you do the Punnett square, they can pass down the recessive condition. There's a 25% chance. And actually, it kind of works out perfectly that 1 out of 4, just by pure chance, 1 out of 4 has that condition. So the condition that we're talking about here, the filled in square, is a recessive trait. It's recessive. We don't know if it's autosomal or sex-linked. Okay, so from this pedigree, we can't really tell if it's sex-linked or not. Yeah, and the reason why we can't, if it was sex-linked, let me actually show you guys if it was sex-linked. The mom could be X little a or X little a. The dad for sure doesn't have it, right? So he's got to be, he's got to be X big A Y. X big A, X little a, X big A, Y. X little a, Y. So do you guys see that that son, he could be X little a, Y? And then this son could be X big A, Y. All right, so we can't really tell if this is sex linked, but we can definitely know that it's recessive, okay? What about the next one? So what type of inheritance pattern is this? So you guys are going to see questions like this probably on the AP exam and then probably on my test as well. Like I'm going to give you like pictures of pedigrees. How do you know, like, I mean, what type of inheritance pattern is this? Is this autosomal? Is it sex linked? Is it recessive? Is it dominant? Okay. Here we have two parents that do show the condition, that have whatever trait we're talking about. But then they have two kids that don't have it, and then they have two kids that do. This is how we know if it's sex-linked or autosomal. You know, when it's sex-linked, it's usually found mostly in males, okay? So here, I can see here that the dad has it, but then the daughters do not. The males do not have it, so it's not sex-linked. We're going to go with autosomal for this one because we don't see it mostly in males. The reason why we would see it mostly in males is because, remember, males only have that one X chromosome. So whatever they get, you're going to see that condition. That's why you see in sex-linked traits mostly in males. But this is obviously not a sex-linked trait because it seems like the girls actually have it. Okay. All right. The question is it dominant or recessive then? Well, it looks like the parents have it, some of the kids have it, most likely it's dominant because two of their kids do not have it. So that means that the parents must be heterozygous. Okay, so if you do a Punnett square, this trait that we're talking about is a dominant trait. There's a 75% chance of showing this dominant trait, okay? And it just so happens that 2 out of the 4 kids, every time they have a kid, there's a 75% chance of having a kid with the dominant trait. And then the recessive trait are these kids, the recessive trait. They just happen to not have the trait, okay? So I guess the easiest way to figure out if it's dominant or recessive is if the parent has it and the kids have it, you know it's going to be dominant. If the parents have it and the kids have it, it's going to be dominant. Because if you go back to this one, the parents do not have it, but the kids have it, then you know it's recessive. Okay, so this is what a recessive pedigree looks like. Parents don't have it, but some of the kids do. This is a dominant pedigree because the parents have it and also the kids have it. Okay? Okay, what is the probability that individual C1, okay, so these are the generations, like this is generation A, this is generation B, this is generation C, this is generation D. It's asking about this one. What's the probability that individual C1 is big W, little w? So in order to do that, we kind of have to do a Punnett square with the generation A first. So we have big W, little w, little w, little w, big W, little w, little w, little w. And so when you do that, The ones without the condition are going to be little w, little w. So this is a little w, little w, little w, little w, little w. Okay, but then person number 2 has to be big W, little w. Do you see that? Because the condition, in this example, the condition is a dominant trait, right? And we know that because it's given to us. It's dominant. But then if they didn't give you the genotypes, you could still know that it's dominant because the parent has it and the kids have it. Okay? So this is the parent here, and then the wife or the female here does not have it, so she's got to be little w, little w. So if you do a Punnett square of generation B here, so the question is, what is the probability of C1, the C1, this guy, being big W, little w, right over here? 2 out of 4, right? So the answer is 2 out of 4, okay? Okay, a typical pedigree for a family that carries Fanconi, I think that's how you say it, Fanconi anemia. Note that carriers are not indicated with half-colored shapes in this chart. Sometimes you might see a pedigree where the carriers are indicated by having a half circle filled. So like this would be a carrier, like X little a. And then if it's completely filled in, if it's a recessive trait that's being analyzed, it would be little a, like that, okay? But in this case, it's just saying that carriers are not indicated. If they have it, then it's going to be filled in. If they don't have it, it's not going to be filled in. So like, for example, George could be a carrier, but he doesn't show it. They didn't do the half circle. Oh man, I'm trying to find my pen. Okay, note that carrier, okay, blah, blah, blah. What is Arlene's genotype? Arlene's genotype. So Arlene has it. She has it. Some of her kids have it. Oh, but you know what? They do give you the genotype. So that's very convenient. So Sam has big F, little f, and he does not have the condition. That means that the condition is recessive. Okay? So that means it's recessive. So that means Arlene is little f, little f. George must be big F, and we don't know if he's big F, or big F, little f yet. Okay? I mean, we could probably figure that out if we just did the Punnett square, right? But let's answer these questions. What is Arlene's genotype? Little f, little f. What is George's genotype? Well, it seems to me that they have a daughter, Wilma, that has it. Son, Tom has it. What is the only way, so that's Arlene right there. What's the only way that George and Arlene are going to have kids that are little f, little f? If George is heterozygous. Okay, so George has to be big F, little f. What are Anne and Michael's genotypes? Okay, Anne and Michael. So Anne doesn't have it, but if you look at the pedigree for her parents, right, this is the pedigree, she's got to be big F, little f. She has to be heterozygous. So you guys, George is a carrier for this. Anne is a carrier for this, as is Sam. They all carry this gene for the condition of Fanconi anemia, okay? Anne is a carrier. Michael, we don't know about Michael, okay? But, however, their daughter does have Fanconi anemia, little f, little f. If Anne and Michael do not have it, but their daughter does, what must be Michael? He's got to be big F, little f, okay? I hope you guys see why. Because if they both do not have it, but their daughter does, that means that Michael's got to be a carrier, just like Anne, okay? Most likely, Sandra's genotype is... Okay, so Sandra is over here. And they have 2 kids. Sandra and Tom have 2 kids, Daniel and Alan. Daniel is big F, little f. They didn't give you the genotype of Alan, but notice how Alan has a son, Christopher, with Tina, and Christopher doesn't have it. So you can sort of see here that the generations after don't have it. So most likely Sandra is big F, big F. Okay? Because if she was big F, little f, then there could have been a chance. They could have had a chance. It was a 50-50 chance of having a kid with the condition, Fanconi anemia. Okay, and you kind of get it. Nobody below them has the condition, or Alan, for example, has a kid who doesn't have the condition. It doesn't say what Christopher is. So most likely, Sandra is big F, big F. Okay. List 3 people from the chart other than George who are also most likely carriers. Okay, so we said George is a carrier. We said Michael's a carrier. Anne's a carrier. Sam's a carrier. Daniel's a carrier. Okay, so those are the people that are carriers. What is the most likely mode of inheritance for this pedigree? You guys remember what I said? It's going to be recessive if the parents do not have it, but some of the kids do. It's going to be dominant if at least one parent has it and then the kids have it. So to me, this is going to be a dominant situation. So I'm going to go with dominant. Okay. But then the other question was, is it autosomal or is it sex-linked? Remember what I said about sex linkage? It's going to be found mostly in males. Male has it, male has it, male has it, male has it, but also females, 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 females. So I'm going to say that this is autosomal because it's not heavily in just one sex or the males. Okay? So I'm going to go with autosomal dominant is the most likely mode of inheritance. So whenever you see that question, what's the mode of inheritance? It's asking you, is it autosomal or is it sex-linked? And to figure that out, if you have mostly males that have the condition, it's sex-linked. If it's a mix of both females and males, it's autosomal. Dominant or recessive? That's the next question. How do you know? It's going to be dominant if one parent has, at least one parent has it and some of the kids. It's going to be recessive if none of the parents have it, but some of the kids do. Okay, that's how you can decide between dominant and recessive. What are the genotypes of individuals 1 through 5? So let me get my eraser again. Actually, maybe I'll just keep that up there. Okay. So we're talking about, we said this is dominant and then this is autosomal. So I'm going to circle these so I don't get confused. Dominant and autosomal. Genotypes of individuals number 1 through 5. So here's the thing. This dad has it. It's a dominant condition. Right? But then you know what? He also has some sons and a daughter without it. And since it's dominant, he's got to be heterozygous. His wife is homozygous recessive because the condition we're talking about is autosomal dominant. So the ones that are filled in, they have this autosomal dominant condition. The ones that do not have it are all going to be little a, little a. Okay, they're not going to have it at all. Okay, so individual number 1 is going to be little a, little a. Let's try to figure out 2, 3, 4, and then 5. That's the question, right? So where's 3? Oh, this is 3. Oh, you know what? This is pretty easy. If it's not filled in, then we know that it's little a, little a. Where's 2? Okay, 2 has to have at least a big A because it's a dominant condition. But then what is the other gene that he has? Well, look at the kids. Little a, little a, little a, little a. So actually person number 5 is also little a, little a. How did this kid got a little a from mom, and then the other little a must have come from dad. So person number 2 has to be little a, big A, little a. Person number 4 got the big A from dad, little a from mom. Okay, big A, little a. Okay, so that's the answer for those set of questions. Next one, and then I think we only have a couple more, and then we're going to be done for today. Done for this video. I think it's going to take hours to export and upload. But anyway, the following human pedigree shows a family affected by a specific disease. Assume, let me move my head. Assume that the individuals marked with an asterisk do not carry any allele associated with the affected phenotype and that no other mutation spontaneously occurs. Basically, it's saying that this person with the asterisk, where is it? Oh, right over here, does not carry it, no family history of it, doesn't have a spontaneous mutation of it, okay? All right, state the most likely mode of inheritance for this disease. I can already tell just by looking at it. See this generation right here with their kids? The parents do not have it, but their kids do. What does that tell you? It's recessive. Okay. Is it sex-linked? Female, male, not sex-linked, autosomal. So we already figured it out, guys. That was pretty easy. That was a really easy one. Okay. So we're going to go with autosomal recessive. So the answer is this guy right here, autosomal recessive. Okay. Write all the possible genotypes for individuals 1, 2, and 4 in the pedigree. Okay, I'm going to go ahead and erase that bit here. Okay. By the way, I mean, some of you guys might find this to be really interesting and fun. I hope you do. So it's recessive. We kind of have to work our way backwards because if it's autosomal recessive, that means that these two individuals have it. If they have it, their parents must be carriers. Their parents must be carriers for it. Okay? So individual 1 does not have it. Also, but then, I'm just trying to think. They could be heterozygous, right? But we don't really know because, no, actually they have to both be heterozygous because if it was big A, big A, if it was big A, big A, then this could, and then if it's little a, then they would have it. So it's got to be like this. It could be like this, or it could be big A, little a. Right? Yeah, it could be either or. So person number 1 is going to be big A, little a. Person number 2, it depends though. It really depends on, write all the possible genotypes. Okay. And then big A, little a, big A, little a. So person number 2 could be big A, or big A, little a. We don't know. There's not a whole lot of information about them. So it's really kind of hard to decide. 1, 2, and 4 in the pedigree. 4. So this dad has it. Mom doesn't. So 4. Mom could be big A, big A, big A, or big A, little a. So 4 could be, if you do the whole Punnett square, 4 could be big A, big, yeah, oh wait, no. It could be big A, no, it has to be big A, little a. Sorry guys, I'm getting a little bit. 4 is definitely big A, little a, okay? Because the dad is little a, little a. Okay, and then number 5, here's the thing with number 5. With the asterisk, they said there's no history of it, blah, blah, blah. So they're trying to tell you that it's got to be big A, because there's no history of this allele in their family. So it's big A. So then if it's big A, little a, then number 5 doesn't have it, could be big A, or big A, little a. Okay? What is the probability that individual 5 will be a carrier? And we determined already it's big A, or big A, little a by 50%. Right? There's a 50% chance of being a carrier right over here. Okay? 50% chance of being a carrier. The following human pedigree shows a family affected by a different disease. Assume that the individuals marked with asterisks, blah, blah, blah. Okay, asterisks do not carry any allele associated with the affected phenotype and no other mutation spontaneously occurs. Also assume complete penetrance. State the most meaning like not, there's no codominance or incomplete dominance. State the most likely mode of inheritance for the disease. I can tell already right away that if you look at this generation, The parents don't have it. One of their kids does. Recessive, right? All right, next one. Look at boys versus the girls. 4 boys have it. Only 1 girl does. That kind of tells me that there is a chance that this could be sex linked, okay? I'm going to go with, let's pretend that it's sex linked. Let's see what happens. So one way to figure this out is over here. The dad has it. Sex linked, recessive, right? So let's say X little a y. The mom, we don't know what the mom is. The mom could be X big A, she could be X big A, or she could be X big A or X little a. We're not sure. Okay. The mom here could also be X big A, or X big A little a. We don't know. But her son, oh, you know what? She's got to be X. You know what? Never mind. This mom is definitely heterozygous because she passed down that X little a to her son. Gotta be. Okay? So that means that this daughter could be, and then this is the dad, X big A Y. The dad is X big A Y for sure because this is a recessive trait. So that this daughter could be X little a, or X little a, X big A, okay, you guys, hold on. This daughter right here, she could be X big A, or X big A, X little a. But take a look at her kids. Her son has it. Her daughter has it. So what does that say? She's got to be heterozygous. Okay? Just to make things a little bit clearer, maybe I can go ahead and show a Punnett square of those parents at the very top. So you know those parents at the very top? Let me just draw a pedigree or a Punnett square because it looks very messy. If you do, I hope that this video is helpful, you guys. I hope that I'm not like wasting my time or your time. Okay, so their daughter does not have it, but she could be X big A, or X big A, X little a. But then her son has it, and her daughter has it. So that means she must be heterozygous. She's got to be this one, not this one. Okay? Wow, lots of X's everywhere. Okay, so then let's do a Punnett square for this couple. Okay, this couple right here. So we just determined that this daughter is X big A. Sorry, you guys. There's some hammering sounds outside. X little a y. X little a, X little a, X big A y, y, X little a y. So this is the Punnett square that shows you guys that the son, this is the son right there that has that condition. This is the daughter that has that condition. So I think this pretty much confirms that this is a sex-linked trait and it's recessive. Yeah. Okay, so we're done with that one. Oh, yay. Okay, so we're almost done with the PowerPoint now for the fun stuff, okay? Because there's no more like practice problems to do. This is a pedigree showing you the royal hemophilia pedigree. If you had me as a teacher for honors bio, you've already seen this. But this is Queen Victoria, you know, famous Queen Victoria, like the Victorian age, right? She was a carrier for hemophilia. So hemophilia, again, is a recessive trait. So normal is the dominant. Hemophilia is X little n. She was a carrier. She had how many kids? 1, 2, 3, 4, 5, 6, 7, 8, 9. She had 9 kids. 2 of her daughters, Alice and Beatrice, they were carriers. Leopold is really famous. Okay, Leopold was the son that inherited the X little n. He inherited the hemophilia gene. Okay, you know, I just remember that Queen Victoria, she actually married her cousin, right? Because her mother is the sister of her uncle, right? They're related. So Queen Victoria married her cousin, Prince Albert. with royal families, there's a lot of like marrying in the family, right? That was a very common thing. And then Leopold had a daughter, Princess Alice, that was also a carrier. And then she had a son with hemophilia. So Viscount, whatever his name is, was X little n y. Maurice was X little n y. Leopold here was X little n y. Beatrice was X big N, X little n. Leopold was X little n y. Alice X little n, X big N, X little n. And then I also wonder, like, if you can tell this is sex linked, because who has hemophilia? All the boys. The boys have hemophilia. And you can see in this pedigree that they do the carriers thing where it's like half filled in. Remember I was telling you guys about that? If it's half filled in, they're carriers. But not all pedigrees do that. Here's where we are right now. We are... Where is it? Oh yeah, Queen Elizabeth. And her husband, Prince Philip. They're actually distant relatives too. Because Prince Philip comes from this line, from Auntie Alice, and then Queen Elizabeth comes from King George, who was descendant of King Edward VII. So they're like really distant cousins. Okay? And then here we have Prince Charles. Princess Diana passed away many years ago. And then you have Prince William and Prince Harry, who now lives in California. So anyways, that's the British British plus Prussian and Russian and Spanish coming together. Okay, we're going to skip that. There's no way that we're going to do that one. Okay, and then here's a very interesting pedigree. I wonder if you guys are going to get to this. And this is definitely not a pedigree like your own. I wonder if I should just save this for class. I might just save this for class. No, it's interesting. Okay. First of all, this pedigree is not like anyone that I've shown you so far. This is a pedigree showing you a connection between a female and two males. Traditionally, I've only shown you pedigrees where it's between one male and one female, right? But in this case, it's a male with two males. And then, so she has two kids, two sons. A son with the first guy and then a son with the other guy. And then this is where it gets really complicated, right? Her first son has a son. Her second son has a daughter. And then the reason why there's two lines is because the two lines means that they're related and also they're joined together. So they're related and then they also got married. And then they had a son and a daughter, and they are also, they're brother, sister, right? They're related, and they also got married. So brother, sister had a son. This part is pretty traditional. Oh my God. Okay, so it kind of jumped. So there's that son where we left off. The son and this lady have a daughter. The daughter is related to her brother, but also marries her brother. And they have a daughter. That daughter is related to her uncle, and they get married. They have a son and a daughter who is also related, and they also get married. So I have to say, this is a real pedigree. I forgot to say that at the very beginning. This is a real pedigree, okay? Actual people. And then, so the daughter and her uncle, they have two kids, a son and a daughter who are brother, sister, also get married. They have two daughters. Daughter marries an unrelated guy, which leads to Cleopatra. Okay, so this was Cleopatra's line. And as I said before, with royal families, they tried to keep it in the family, tried to keep the royal line going. And so that kind of explains that phenomena where people are marrying a relative, okay? Wow, this was a really long one, wasn't it? Well, if you did watch the whole thing, I'm impressed. I hope it was helpful. I hope it's not a waste of time. And so thank you for watching, and I hope it was helpful.